it.
All right, greetings to everyone. First and foremost, I want to extend my profound gratitude to the organizers of this Congress, and I wish everyone the very best of luck. Elisha Apetewe Nakambong is my name, and I'm presenting a portion of my research work entitled An In Vitro Investigation of the Interaction of Genomic DNA with Copper Chloride under the supervision of the second author. First and foremost,
Dear Dr. Alisha, we cannot hear you. I think there is an internet connection problem. If you allow Dr. Şebnem, we can continue with the next one.
Okay. Okay, we can continue with the next one. I think there's an internet problem. The next presenter is Dr. Ramazan Kurul. He will present investigation of kinesophobia, proprioception, state, and trait anxiety levels in geriatric patients with shoulder injury. Dr. Kurul?
Thank you. Can you hear me?
Yes.
All
We
right.
can hear you.
I would like to thank the Congress board and the chair for the successful Congress. I was also part of the first one, and first one as well also was great. I directed 3 of my students to the oral presentation in this one. This is a really successful and well-organized Congress. I would like to thank you. I think I am successfully shared my screen, right?
Actually, we are not able to see full screen.
Is it full screen now?
In your computer, yes, but for the audience, no. Just go to upside of your presentation now, then press the green button and reselect the slideshow, not the poster presentation, slideshow.
Right. How is it now?
Now we can also see your next slide.
Next, just maybe I can.
Perfect.
All right. The topic of my presentation is investigation of kinesiophobia, proprioception, state and trait anxiety levels in geriatric patients with shoulder injury. My presentation will be starting with background, then aim, method, results, and conclusion. As a background, the rotator cuff is a group of muscles and tendons that surround the shoulder joint, keeping the head of the humerus firmly within the shallow socket of the shoulder. A rotator cuff injury can cause a dull, aching pain in the shoulder, which often increases with the usage of the arm. As you can see, there are a lot of muscles that are responsible for the stability of the shoulder, and it is really easy to any one of these muscles get injured with excessive motion or with age. Rotator cuff injuries are common, and the ratio of these injuries increase with the age. Elderly people prone to secondary shoulder injury after the initial lesion, because after the first lesion, as you can see, there are all of these muscles are responsible for the shoulder's suspension. The first one is deltoid, and the second one is the supraspinatus muscle. Usually the supraspinatus muscle gets injured and it loses its function. Then the deltoid take over its function. However, its strength and angle of force is different than the supraspinatus muscle, which results the increased pressure of the head of the humerus towards the acromion, which compresses even further the supraspinatus muscle, which increases the lesion. Also, with inactivity, the elderly people are prone to atrophy, muscle atrophy, which they are more susceptible to the younger people, because they are in risk of the sarcopenia, which increases the ratio of the shoulder injuries among the elderly people. Many people with the rotator cuff disease can manage their symptoms and return to activities with physical therapy exercises that improve flexibility and strength of the muscles surrounding the shoulder joint. Early rehabilitation is effective, and according to reviews, it needs to be performed. However, due to the COVID-19, elderly population became more immobile and their accessibility to rehabilitation is reduced. That increases the reported kinesiophobia, pain, and limitation in daily life activities among the elderly people. Therefore, aim of this study was to investigate the effect of kinesiophobia, proprioception, state rate anxiety levels of geriatric shoulder injury patients, and investigating relationship between kinesiophobia and anxiety levels in accordance with the perceived pain scores. A total of 24 individuals, 13 female and 11 male, with a mean age of 70.33 years, who diagnosed with unilateral soft tissue shoulder injury were included in this study. Individuals' mean onset of shoulder injury was 6.5 months. Laser-pointed angle reproduction test was used to assess the proprioception. Functional status levels of the patients were evaluated with constant Merrill scoring. International Physical Activity Questionnaire was used to assess the physical activity of the elderly people. Also, Tampa scale for kinesiophobia were used for assessing the kinesiophobia. Application of state rate anxiety scale was performed with visual analog score, which assessed the perceived pain levels. As for the results, constant Merrill score and Tampa kinesiophobia scale scores with state and rate anxiety scores showed a significant difference between groups with and without soft tissue injury, as a result of Spearman's correlation analysis that showed a strong relationship between constant Merrill scoring and visual analog scale scores. Also, there was a significant difference between groups with and without soft tissue injuries in terms of functional stationing, cognitive functioning, anxiety, and kinesiophobia. There wasn't a significance between groups with and without soft tissue force, proprioception, and physical activity levels. For conclusion, according to our results, geriatric patients develop kinesiophobia and level of the anxiety increases with increase of perceived pain. Rehabilitation needs not to only focus on shoulder functions during the treatment process in geriatric patients, but also establish a treatment process by considering anxiety, kinesiophobia, and cognitive stress. That concludes my presentation. Thank you for your time.
Thank you very much, Dr. Kurul. This is a very nice presentation. The audience, do you have any contribution or question? Okay, thank you very much, Dr. Kurul.
Thank you.
Okay, the third presenter is presentation belongs to Dr. Sümeyye Başar. She's going to present us determination of antibacterial activity of boron compounds against the cause of hospital infection Pseudomonas aeruginosa, ATCC 27853. Okay, Dr. Başar.
Full screen.
Could you please do it full screen? First, you have to open full screen. After that, you will see green button. First, you have to open the presentation in the full screen in your laptop, and then after that, you will go to the Zoom and see the green button. Yes, Thank you.
Dear participants, welcome. Today I will give you my oral presentation on determination of antibacterial activity of boron compounds against the causes of hospital infections, Pseudomonas aeruginosa, ATCC 278, 5-3. Pseudomonas aeruginosa is a common encapsulated gram-negative strict aerobic rod-shaped bacterium that causes disease in plants and animals, including humans. A species of considerable medical importance. In addition, biofilm formation and inflation detection system induce adaptive resistance during bacterial growth. This leads to multi-drug resistance strains, particularly resistance to carbapenem. Infections and spread of resistant microorganisms lead to high morbidity and mortality. In this context, with the development of multiple resistance to antibiotics, alternative treatment methods are sought. Studies on boron compounds have examined boron application from material used to medicine. Boron-based drugs have a variety of uses, including imaging agents for both optical and nuclear imaging, as well as therapeutic agents with anti-cancer, antiviral, antibacterial, antifungal, and other disease-specific activities. Bacterial stream of fresh 24-hour culture was prepared by passage of Pseudomonas aeruginosa strain, L-methylene blue and blue agar. Bacterial suspension was prepared from the colonies taken from the prepared cultures in saline with a turbidity of 0.5 McFarland, 5 times 10 to the 5 coloniformic unit milliliter. Preparation of boron compounds. 15 boron compounds were dissolved with double distillate water to determine a final concentration of 40 millimolar, 500 micrograms milliliter. Minimal inhibition concentration. 100 microliter of tryptic for broad medium was added to all wells of the 19-6 well microplate. 100 microliter from wells 1 to 10 of boron compounds were added, and dilution was made up to the 10th well. 100 microliter of preparate Pseudomonas aeruginosa strain suspension was added to all wells. Bacteria control was added to 11th well, and only the bacterial suspension and only boron compounds were added to the 12th well. It was incubated for 24 hours at 37 degrees Celsius. Wells showing inhibition were not. In our study, antidote, sodium metaborate tetrahydrate, sodium tetrafluoroborate, and Lexis didn't show any antibacterial activity, while sodium perborate monohydrate 62.5, boric acid and ammonium tetra fluoroborate 31.25 microliter, calcium metaborate 15.6 microliter, and zinc borate, ammonium pentaborate tetrahydrate, sodium terfluorine borate 7.81 microliter showed antibacterial activity. Various boron-containing compounds have entered the literature as biological activity molecules in recent years. In the light of this information, calamite, ammonium pentaborate, tetrahydrite, zinc borate, and borax showed the most effective antibacterial activity on Pseudomonas aeruginosa strain. However, the antibacterial activity efficiency at higher doses should be tested, and non-toxic dose range should be determined by experimental models and cell cultures in vivo studies. Thanks for listening to me.
Dr. Bashar, thank you for your nice presentation. And to the audience, do you have any questions, or does anybody has contribution? Okay, thank you very much. And we have solved the first presenter's technical problem. And now I would like to announce him. First speaker was Elisha Apatevan Akampong. He's going to present us an in vitro investigation of the interaction of genomic DNA with copper chloride.
Hello. Please, can you hear me?
Yes, yes, we can hear you.
All right, a quick recap from the previous slide. As I was saying, DNA is a genetic nucleic acid material that is essential for RNA synthesis as well as protein synthesis. And it's also essential for cell survival and cell proliferation. Metals and metal cations such as potassium, sodium, and magnesium are essential for the cell normal functioning of a cell and also for the stability of DNA. Thus, they can be referred to as friends of DNA. Also, there are some metals that are known to be mutagenic and carcinogenic. Thus, they are referred to as foes or enemies of DNA. And facts regarding the mutagenicity and carcinogenicity of these metal ions were gathered from various studies conducted to investigate the interactions existing between D block elements and DNA. Copper complexes, due to their three-dimensional structure, cationic and redox ability, they have the aptitude for interacting with DNA. Thus, they can be referred to as DNA binding molecules. DNA binding molecules greatly influence the normal function of a cell, as they can modify and inhibit the activity of a cell's DNA. And this is a field of research that has gained much attention, essentially in the field of drug development. That is the development of therapeutic agents. They are widely used and accepted. However, there are already existing metal-based therapeutic agents such as cisplatin, but due to the intolerance and toxicity of such therapeutic agents, there is the need for the development of more effective and safer therapeutic agents. And to do this, the interaction of DNA with metal complexes has to be exploited. In spite of this, there are less studies investigating the interaction of DNA and metal complexes. Thus, this study aims to add to existing literature by investigating the interaction of DNA with copper chloride. This study was conducted in the molecular laboratory of the Department of Biochemistry, Kirkcaldy University. And in this study, a DNA with a purity of 1.84 isolated from calf thymus was used for this study. And calf thymus tissues were the main source of the DNA because calf thymus tissue is a rich source of nucleic acids. Also, purchased copper chloride was prepared into 5 different concentrations, that is 1,500, 250, 125, and 62.5 micromolar. Following that, combinations were made with the isolated calf thymus DNA in 0.2 ml sterile Eppendorf tubes. After the combinations were made, the combinations were incubated at a temperature of 37 degrees Celsius for 60 minutes. After incubations were successfully done, all the samples were now red, or the absorbance intensity of the samples were measured using a UV, an ultraviolet visible absorbance spectrophotometer. And in the measurement of the absorbance, only DNA was measured as a positive control. Only copper chloride, that is the various concentrations of the copper chloride, were also measured as the negative control, and the combinations as well were measured. As expected, the control, that is the positive control, the calf-thymus DNA gave an absorbance intensity at wavelength 260. Also, as expected, the various concentrations of the copper chloride did not give any absorbance intensity at wavelength 260. Now, with this is the graph representing the combinations, that is calf-thymus combined with the various combinations, with the various concentrations of the copper chloride. And if it can be observed, aside from the control DNA, the combinations also gave a peak. And inferencing from the previous slide, where only the various concentrations of the copper chloride were measured, there was no absorbance intensity at wavelength 260. Hence, the observance or the manifestation of absorbance intensity, the absorbance at wavelength 260 is an indication that the copper chloride actually bind with the calf-thymus DNA. If this has been established, that is the fact that the copper chloride has bind to the calf-thymus DNA has been established, then the next question is, by which mode did this copper chloride bind to the DNA? And generally, there are two main modes that are used to, that are actually used. And these modes are intercalation and group binding. And now, if a DNA binding molecule binds to DNA by intercalation, there is always the manifestation of what? Hypochromism. That is to say that as the concentration of the binding molecule increases, the absorbance intensity decreases. Also, if the DNA binding molecule binds to DNA by group binding, then there is the absorbance of hyperchromism. That is to say that as the concentration of the DNA binding molecule increases, the absorbance intensity or peak also increases. And that was observed in this case. If it is observed keenly, 62.5 micromolar concentrations of the copper chloride when interacted with DNA, the absorbance peak given was lower as compared to that of the 125, and that of the 125 was also lower as compared to that of 250, and so on. Thus, it can be said that the DNA, the copper chloride molecule binds to DNA by means of groove binding. Aside from that, one can also extrapolate that the manifestation of increment in absorbance intensity probably was as a result of DNA uncoiling effect instigated by the copper chloride compound. In conclusion, I would like to say that copper chloride has the aptitude for interacting with DNA. That is to say that it can bind with DNA. And from the results of this study, it is inferenced that copper chloride binds to DNA by means of group binding. Hence, it can be used in the development of therapeutic agents, such as anti-cancer agents, since the increment in absorbance intensity probably was as a result of DNA degradation instigated by the copper chloride. However, I will also recommend that further or subsequent studies should be conducted to investigate the interaction of DNA with copper compounds, precisely copper chloride, and the possible DNA cleavage activity that can be incited or instigated by these copper compounds. Thank you for your audience.
Thank you very much, Dr. Elisha. I want to ask something. If we use copper chloride for the therapeutic agents, so there will be a decrease in the costs of the therapeutics or not? Is it? I think it's cost effective, right?
No, cost-wise, it won't be cost effective because already there are existing metal-based therapeutic agents, like I stated, cisplatin. But these existing metal-based therapeutic agents also have their side effects. So this study only sought to add to existing literature to propose another an alternative material or method that can be used in drug synthesis.
Okay, thank you very much.
You're welcome.
To the audience, do you have any contribution? Okay, thank you very much. And the next speaker is the Dr. Aklilu Abraham Roba. They are going to present, is this by chance or an iceberg? A case report of gastroschisis from Kayseri.
Thank you very much, Mr. Chairman, and also the organizers. Can you hear me?
Yes.
Okay, thank you. Is this by chance or is it an iceberg that shows a cumulative incidence of gastroschisis in the public or in the community? So I will present a case that we have seen in hospital during hospital visits. That's a gastroschisis. Gastroschisis is a rare congenital malformation. That's the protrusion of the intestinal organs through defects in the anterior abdomen, usually to the right side of the umbilicus. And usually intestine is protruded, but sometimes organs, other organs such as the stomach and the liver also can protrude through that. And when we see the survival rate is around 90% in industrialized countries, whereas in developing countries or in poor industrialized countries, the death rate is around 100%. So intensive medical care is very important. And the prevalence is increasing globally and also in regions. For example, in the regions that Turkey located in the Europe, it's also increasing, but it varies from countries to also countries. For example, in UK and Wales, the prevalence is highest, and in Italy, it might be lowest. Some literature says like that. And the exact population-based magnitude and factors in Turkey I haven't seen in the published literature, so it might not be exactly known. But in a single week, in December 2021, we have seen 2 units born with gastroschisis from a young mothers in a university hospital, Erciyes University. So therefore, this caught our attention to investigate the possible risk factors based on one of the cases presented. I will present one of the cases. The baby was delivered on December 26 with cesarean section due to premature rupture of membrane at 32 weeks of gestation. He was boy, and 1,600 grams, 1.6 kilogram, its lower weight. And the baby was healthy, except there is protrusion of intestinal organs. And one thing that complicates the baby is the gastroschisis was meconium stained. So medically that complicates the conditions. The mother was 20 years old. She's gravida 1, and she has no marriage with relatives. She has no chronic diseases, preeclampsia, or other genetic diseases in the families, but she smokes. And the baby was admitted to neonatal intensive care unit, and surgical team placed the exposed intestine into a silo bag, and so it was served and suspended above the infant to allow the intestine to slowly reduce over time, and then after to perform surgical operation. During physical examination of the baby at the neonatal intensive care unit, almost everything is normal except there is opening in the abdomen, that's gastroschisis, and the baby was initially the baby was at the bad condition because of prematurity, lower weight, as well as the gastroschisis was meconium stained. So with treatment, there are medical and nursing management. The baby was on radiant warmer. He was received 10% dextrose and different antibiotics, vitamin K, vitamin D, as well as pain medications, and also cream was applied around the silo. and also decompression was done with orogastric tube to reduce abdominal pressure. The following things are very closely monitored with charts: all fluid intake and output, vital signs, blood chemistry, blood gases, electrolytes, and acid base balance were daily closely monitored. With nursing management, one of the important areas is skin integrity. So the baby has altered skin integrity due to disease process, as well as protrusion of abdominal content, and also risk of tissue damage by invasive procedures. So as a plan, wound care was planned and it was implemented. The other and most important component is feeding. Parenteral feeding was initiated. Fluid volume is also another important component because of insensible water loss is there. So rehydrating the baby was important, and it was also done. The other breathing problem, the baby was low birth weight as well as preterm. So there is ineffective breathing pattern related to prematurity. So monitoring respiratory rate and breathing efforts were planned, and intervention was done. The other important component is pain management. Because of the nursing procedures, disease condition, and surgical management, the child has expected pain. So administering anti-pain, using pacifiers, massage, and music therapy as a plan, it could be done. Infection prevention is the most another important component because of risk of surgical site infection related to invasive procedures, and also security and safety is also another important component that are related with nursing care. With risk factors, in body of literatures, there are a lot of risk factors are mentioned, but for our particular mother also, the mother is young age. So in the body of literature, younger maternal age is one of a risk factor, and also maternal smoking and alcohol use was one of the known risk factor in the body of literature. Our mother didn't drink alcohol, but she smokes, so maternal smoking is one of a risk factor. And in the recent literatures, exposure to wildfire smoke is one of the risk factor for gastroschisis, and Turkey is one of recent years exposed for wildfire. So our mothers also might be exposed for wildfire, maybe. So maternal smoking and exposure to smoke can be considered as risk factors. So with conclusion and recommendations, young maternal age, environmental exposure to smoking and wildfire were listed as possible risk factors. Therefore, programs that reduce maternal exposure to smoking should be encouraged, and also we recommend studies with strong design and also larger geographic areas. These are my references. Thank you very much.
Thank you very much. Luckily, we diagnosed gastroschisis and omphalocele during prenatal period with obstetric ultrasound. And gastroschisis is not with chromosome abnormalities, but I will ask something. What type of music do you use for to lower pain?
Yeah, normally in the hospital, they didn't apply that music intervention, but there are teams that use music interventions for neonates to distract them from pain, and that was effective in reducing pain. Yeah.
That there is a list, I think, in psychiatric society's page that affects the pain. Yes. Okay. Thank you very much.
Thank you.
And the next presentation is from Dr. Hadoi Hajar. They are going to present us obesity, prevalence of hypertension and metabolic syndrome in a woman sample from Koribga, Morocco.
Thank you. Hello, everyone. Honorable assistants, today we will present this project to you as obesity, prevalence of hypertension and metabolic syndrome in a woman sample from Khouribga in Morocco. Obesity and
Wait,
access.
do you have slides?
Yes.
I could not see them. Could you share your slides?
Sorry.
Could you share your slideshow?
Yes, yes, it's okay. Okay.
I could not see.
Thank you.
I couldn't see it. First of all, you have to open your slides and then you have to do it full screen.
Nous ne pouvons pas voir votre présentation. Pouvez-vous ouvrir votre présentation?
Comment s'il vous plaît?
Tu peux utiliser du bouton vert.
Vert, ok.
Après, tu dois cliquer votre présentation.
Okay. D'accord, ok. Thank you. Okay. We will present this project to you as obesity, prevalence of hypertension and metabolic syndrome in a woman sample from Khouribga in Morocco. Obesity. This is clear? This is clear?
Yes, we can see it, but you can do it full screen. If it is not possible, it's okay.
Okay. Obesity and excessive accumulation of fat in the body, it can affect individuals regardless of age, sex, income, or country. We have obesity general or abdominal or central obesity. Among causes of obesity, we distinguish environments, genetics, high-calorie foods, and the effects are also multiple. We distinguish cancer, hypertension, insulin resistance, and metabolic syndrome, and psychological problems. In the world, obesity rates have tripled over the past 4 decades worldwide. Currently, obesity can be a global epidemic. Worldwide, an overall prevalence 12% in adults, approximately 604 million obese adults. Obesity affects between 30 and 40% of adults with 8% for morbid obesity in some countries. In Morocco is in demographic, epidemiological, and food transition due to a significant increase in life expectancy and the rate of urbanization. The prevalences evolution of obesity between 2000 and 2017 by gender and residence. Increasing rates obesity is 6%, 8 in 10 years. The objectives, the study, primarily evaluate the rates of general and abdominal obesity, analyze their link with hypertension and metabolic syndrome in a sample of Moroccan women from the province of Khouribga in Morocco. Finally, estimation of lipid accumulation products index predictive of the metabolic syndrome. Cross-sectional survey was carried out in the Moroccan province of Khouribga in 2019. Simple, it's 117 randomly sampled women of 18 to 65 years old. Measurements are weight, height, waist, and hip circumference were measured. Body mass index and weight on hip ratio were calculated. The blood pressure, the blood pressure was also recorded, as well as the following biological parameters: glycemia, total cholesterol, triglyceride assessed in fasting plasma. The equation for lab is the following. Sorry. The all anthropometric measurement means BMI, waist circumference, waist to hip ratio are exceed the norms. The prevalences general obesity, it's 47%, and abdominal obesity, it's 79%, 14. Hypertension and lab better than 53 rates are 54% and 42% and 9% respectively. Khribga samples particularity, its prevalence are high and comparable to Sahrawi Moroccan and Smara and Lyoun and Oujda. In the sample, there are two groups, normotensive and other hypertensives. In hypertensives, the rate of obese women and with morbid obesity are higher. In addition, the prevalence of hyperglycemia, hypertriglyceridemia, metabolic syndrome, and lab better than 53 are higher hypertensive compared to normotensive. Let's compare the mean of all the parameters between the groups with the metabolic syndrome and the other without metabolic syndrome. The difference are statistically very significant, except for total cholesterol. Anthropometric measurements, especially abdominal obesity, is very high in the group with the syndrome. Let's Spearman's correlation confirm the link between obesity and high blood pressure. General obesity and abdominal obesity are correlated with the systolic blood pressure and the diastolic blood pressure. Also, obesity, abdominal or general are positively correlated with the risk parameter metabolic syndrome. Abdominal obesity is a high risk factor for developing metabolic disease. The prevalence, conclusion, the prevalence of obesity in the Khribga region is very alarming. These results show that abdominal obesity is very common in people with the metabolic syndrome. Abdominal or central obesity is the risk factor very important on metabolic syndrome development and other cardiovascular disease. The prevalence of hypertension is high in women with obesity. Metabolic syndrome is a global scourge, and LAP is a cardiovascular disease and metabolic syndrome predictive in our sample. recommendation, I have to recommend the practice regular sporting activity, have a diversified and balanced diet, and limit your composition of saturated fats. And thank you for your attention.
Thank you very much, Dr. Hajar. It was a nice presentation. Obesity is a worldwide problem, really. From the audience, does anybody will contribute? Okay, thank you very much.
Okay, thank you.
And now Dr. Sabina Kanam. She's going to be present a study on malaria prevalence on general population of Damaturu, Yobe State, Nigeria. Dr. Kanam?
Yes, I can hear you, yes.
Could you do full screen? Thank you.
It's visible?
It's okay, thank you.
Okay. So today, my presentation topic is study on malaria prevalence on general population of Damaturu, Yobe State, Nigeria. The background of the study is malaria is a public health problem which causes medical, economic, and social implications. It kills more than 1 million people annually in tropical and subtropical developing countries. Malaria is caused by protozoans of genus Plasmodium, which is an infectious disease of humans transmitted through infected female Anopheles mosquito. There are several methods to control malaria. It can be controlled by performing surveys on local basis and by accurate and prompt diagnosis. These methods can help in finding out the impact of disease on the general population, on the health status of the general population. Such methods also reduces the unnecessary use of anti-malaria drugs for the treatment of malaria, and as drug selection for the treatment of malaria depends on a species of malaria parasite in the malaria patients. There are several studies shows malaria epidemics is associated with some climatic conditions such as temperature, humidity, and rainfall, because in some places the transmission of malaria is seasonal. In rainy season, infection of malaria is more common because of increase in the breeding sites, temperature between 20 degrees centigrade to 30 degrees centigrade, and humidity is of about 60% is the optimal condition for the transmission of malaria. If the relative humidity increases, then the longevity of mosquito increases. The methodology of this study is survey method was used for this study. This method was chosen because it makes it possible to collect data on a large population using questionnaire. The population and sample is the target population for this study consists of patients and the hospital management and medical personnel residing in General Hospital Damaturu, Yobe State, Nigeria. For the data analysis, the data were analyzed with the SPSS to find association of malaria with testis variables, and p-value of less than 0.05 was considered as statistically significant. In results, the first variable general characteristics of the participant. In this study, a total of 178 sample size used. Out of 178, 80 were male and 98 were female. All the participants have been categorized into 6 age groups, that is 1 to 10, 11 to 20, 21 to 30, 31 to 40, 41 to 50, and greater than and equal to 51 years of age. Most number of the respondents falls under the age group 41 to 50, that is 26.25% in males and in females it is 21, it is 37.75% of the age group 21 to 30. It is evident that the females were more affected from malaria than the males in the Maduro. Out of 178 respondents, 21, that is 26.25% males having income between 11,000 to 20,000 naira, and 29, that is 29.59% female having more than 21,000 naira monthly income. Out of 178 samples, 10 personals, that is 12.5% males, were having mud houses. 21, that is 26.25% males, were in concrete houses. 7, that is 8.75% in both mud and concrete houses, and 42 males, that is 52%, gave no response for this study. While 21, that is 21.42% female respondents were living in mud houses, 29, that is 29.59% females were living in concrete houses. 10 females, that is 10.20% females, were living in both houses, and 38, that is 38.77%, gave no response for the study. The next parameter was the prevalence and sorry. Out of 178 participants, 80 males and 90 females females are examined for the malaria. Out of 80 male respondents, 11, that is 100% out of 11, is of age group 1 to 10. 9 out of 13 is the age group of 11 to 20. 8 out of 11 in 21 to 30 age group. 5 out of 17 in 31 to 40 age group. 7 out of 21 in 41 to 50 age group are malaria positive. While in the case of females, 6 out of 6 of the age group 1 to 10, 8 out of 17 of the age group 11 to 20, 10 out of 37 of the age group 21 to 30, 9 out of 26 of the age group 31 to 40, and 5 out of 11 of the age group 41 to 50 were malaria positive. This study shows that the malaria prevalence was highest in the youngest age group, that is 1 to 10. Out of 178, 21 respondents have been malaria positive due to their poor sanitation. 17 were infected due to the stagnant water. 15 were having it due to the low level uses of mosquito net. 11 were infected with malaria due to the both poor sanitation and stagnant water, and 98 don't know the reason of malaria. Out of 178, 67 were visiting hospitals or clinics for the treatment of malaria. 5 were used traditional medicines. 9 were using self-medication. Moreover, out of 178 malaria patients, 2 were used both hospital and traditional medicine for the treatment of malaria. 5 were visited hospitals and taking self-medication. 5 were used all treatment methods, and 98 patients we don't know about the treatment of malaria. Therefore, it is suggested that the local government, state government, and federal government spend more on the awareness of the treatment of malaria in Nigeria. This study also investigates over the prevalence and distribution of malaria among the participants according to their education level and gender. Out of 178 malaria patients, 80 were males while 98 were females. However, out of 80 male patients, 3 having primary education, 14 went up to senior secondary, 27 were obtained BSc or graduation degree, 4 were having MD, and 32 were not educated. Moreover, out of 3, 2 of them having malaria positive with primary education. Out of 14, 6 of them positive with senior secondary education. Out of 27, 11 were positive with BSc degree, and out of 32, 23 had no education. While in the case of females, 2 were obtained primary education, 11 were having senior secondary, 37 were having BSc, 3 of them having MD, and 45 had no education. Moreover, out of 2 female patients, 0 had malaria positive with primary education. This results is quite interest to compare to male malaria patients with primary education. Out of 11 females, 3 having malaria positive with secondary education. Out of 37, 7 were malaria positive with BSc degree. Out of 3 female patients, 0 having malaria positive with MD, and out of 45, 28 were positive with no education. The prevalence and distribution of malaria among the participants on the basis of income and gender. For the analysis of data, monthly income has been categorized into 4 groups of Naira 1,000 to 10,000, 11,000 to 20,000, greater than and equal to 21,000, and with no income. A total 178 patients, 80 males and 98 females sample were taken for this purpose. Out of 80 male patients, 5 males having income of 1,000 to 10,000, and out of these 4, that is 80% are having malaria positive. 21 males fall in Naira 11,000 to 20,000 income group, and out of these 21, 8, that is 38.09% have been malaria positive. 11 males have been tested and 2 were found malaria positive with greater than Naira 21,000 income group. Out of 12, 12 are having positive with no income. While out of 98 females, 4 were tested, 2, that is 50%, found malaria positive with Naira 1,000 to 10,000 income group. 10 were tested and 1 found positive with Naira 11,000 to 20,000. 29 were tested and 6 found malaria positive with greater than or equal to 21,000 income group. 23 were tested and 13 females
Sorry,
are
could you
malaria
please
positive.
summarize? Sorry, could you please summarize? We are late.
Therefore, it is concluded from the above data analysis that there is positive relationship between malaria positive patients and their income. In the conclusion from the above analysis, one can conclude that the federal government, state government, and local government should have to create more employment opportunity for their people so that they get income, and further they may wisely use it to prevent malaria in the region. Thank you.
Thank you very much, Dr. Kanan, for this presentation. The next speaker is Fatima Zahra Kealu. She's going to present study of the correlation between the MAG polymorphism and the AG mutation of the mitochondrial DNA and the type 2 diabetes in a Moroccan population.
Hello. Just 1 minute. I want to share. Hello, you hear me?
Yes, that's okay.
Okay. Thank you, and I have the honor to present to you the main result of my work, study of the correlation between the polymorphism A1039AG and the mutation A3243G mutation of the mitochondrial DNA and type 2 diabetes in a Moroccan population. This presentation will take place according to the following plan. Diabetes is an insidious and progressive chronic metabolic disease that results in a disorder of glucose assimilation, utilization, and storage. This results in chronic hyperglycemia, and monitored or undiagnosed diabetes can lead to many complications. It is a multifactorial disease The multifactorial disease manifests itself in different forms, type 1 diabetes and type 2 diabetes. However, type 2 diabetes remains the most common form, accounting for 90% to 95% of cases. Its appearance depends on environmental and individual factors, such as physical activity, age, gender, as well as nuclear and mitochondrial genetic factors, whose presence of mutation in the mitochondrial DNA is not negligible. In fact, the molecular origin of diabetes that is characterized by a decrease in the action of insulin is not known. A role could be attributed to the mitochondria. Mitochondria is a small intracellular organelle that contains its own DNA, mitochondrial DNA, and play a major role in the synthesis of ATP, has an essential place during many metabolic processes. ATP play a role in the regulation of intracellular signaling cascade. Indeed, many metabolic diseases are the consequence of mutation of mitochondrial DNA, such as diabetes, that can be associated with impaired mitochondrial metabolism, disorganization of the respiratory chain, excessive production of reactive oxygen spice, ROS, resulting in the apoptotic process or programmed death of cells. I have already mentioned that mitochondria intervene during many metabolic processes. Mitochondria play an important role in carbohydrate metabolism and therefore a role in insulin secretion, since the mitochondrial respiratory chain is associated with ATP production and thus ATP-mediated insulin secretion in pancreatic beta cells. As you notice in this figure, an increased production of ATP leads to closure of ATP-dependent K+ channel, and this leads to cellular depolarization that allow the opening of calcium channel and stimulate the release of insulin. And so mitochondrial dysfunction can contribute to the development of diabetes. mtDNA, mitochondrial DNA, encode for 37 proteins, 13 of which encode for the subunit of complex 1, 3, 4, and 5 of respiratory chain. Mitochondrial DNA code for 5 of the protein in the respiratory chain, nuclear DNA for the 95%. The role of mitochondrial DNA in the predisposition to diabetes has been suspected for about 10 years and more. In this study, we are interested of polymorphism A1039AG, which is localized at the level of NADH dehydrogenase 3 of the complex 1, and the pathogenic mutation 3243, generally associated with deafness and diabetes. The main objective of this work is to research and study the association between this polymorphism of mitochondrial DNA and the pathogenesis of type 2 diabetes in a Moroccan population through a case control study. In the regard, I would like to clarify that no work in Morocco has studied the involvement of this polymorphism in the appearance of type 2 diabetes. This research project has been approved by the Ethics Committee, and it was carried out according with the Helsinki Ethics Declaration. Method: the DNA extraction was performed by the kit DNA ecosystem. in order to visualize the PCR product, electrophoresis was performed on agarose gel at 1%. DNA sequencing was performed by Sanger technique in the order to analyze the genetic sequence. A reading of the chromatogram of the sequences was performed on chromas and the genetic analysis on blast. The reference sequence of human mitochondrial DNA was brought from Mitomap. The collect data was analyzed by SPSS software. In this study, 122 subjects were included. 78 patients with type 2 diabetes who had an average age of 16 years and 43 healthy control participants had an average age of 54 years. As we have already mentioned, the objective of the study is the study the association of mitochondrial DNA A1039 Ag polymorphism with diabetes. At the level of figure A, we observed the substitution of adenine to guanine, responsible of mutation for sense, change the amino acid threonine to alanine. In our population, the allelic frequency distribution of this polymorphism so that allele G of allele G was significantly more common in control compared to diabetic. In contrast, the frequency of allele A was significantly higher in diabetic compared to the control. Sequencing results for the pathogenic mutation 3243 in our population, including the 3 diabetic subjects, the result revealed the absence of this mutation. These results are in line with those achieved in Pakistan, Tunisia, and Indonesia. However, some study conducted in Japan revealed the presence of this mutation. Our studies show an association between diabetes and this polymorphism. These results are consistent with those of several studies, including that of Sharma et al., showing a significant association between this polymorphism and type 2 diabetes in Jammu region of India, whose allele A is more common in diabetic, while allele G is very common in control. These figures show that allele A can lead to decrease the electron transport chain efficiency and increase electron leakage at this site. This results in increased mitochondrial and cellular growth, which can lead to mitochondrial dysfunction and nuclear DNA mutation. Previous studies have shown that overproduction of ROS related to the allele A decreased insulin gene expression. Increased ROS production may also lead to a production in ATP formation, leading to impaired insulin secretion. We suggesting that the allele G has a lower potential to generate the ROS. Our study show a significant association between the polymorphism A1039-8G and diabetes. The allele A is more common in diabetic. Our studies suggest that this allele may be a marker of potential risk of susceptibility to diabetes. The allele G is more common in control. That suggests that this allele may have a protective effect on diabetes. Absence of the pathogenic mutation A3243G in our population, including the three diabetic, may not be the main cause of deafness in diabetic study. To confirm this finding, large-scale study and dependent deafness-related mutation in the nuclear genome may be needed. Finally, I want to express my gratitude to every contribute to realize this work, and thank you for your attention.
Thank you for your nice presentation. It's a very valuable research. I hope it will be an original article soon, and it will contribute to the literature. Thank you very much. From the audience, is there any question or contribution? Okay, thank you very much.
Thank you.
And the next presentation belongs to Dr. Salahattin Redon in silico screening and exploration into phenotypic alterations of deleterious non-synonymous single nucleotide polymorphism in CYP A1 gene. There's a problem with your voice. Voice.
Çıktı değil mi?
Could you please do full screen?
Il y a un problème avec votre voix.
Could you please open your voice? We could not hear you. I think there's a problem with your computer.
Nous pouvons continuer avec la présentation prochaine. Tu peux hocam devam edelim biz diğeriyle.
Okay, we will continue with the next presentation. The next presentation belongs to Dr. Bushfia Amale. The presentation is genetic cause determination of some syndromic deafness by whole exome sequencing in Moroccan families.
Hello.
Hello, could you do full screen, please? Yes, we can hear you. Could you do full screen?
My screen is, can you see my screen?
Yes, it's okay now. Thank you.
Excuse me.
Okay, thank you.
I'm very happy to participate with you at this Congress. At first, I congratulate the organizers of this Congress, and I thank them warmly for the opportunity they give us to present our work entitled Genetic Cause Determination of Some Syndromic Deafness by Whole Exome Sequencing in Moroccan Families. This study was carried out in the Laboratory of Genomics and Human Genetics of the Pasteur Institute of Morocco in the framework of the project to determine the molecular basis of hereditary hair loss in Moroccan patients. Hereditary hair loss is the most common sensory disorder. Approximately 2 per mil newborns are affected. In children, this affection has dramatic effects on language acquisition, educational progress, and daily lives in general. In adults, it causes social and professional problems. This monogenic affection is of great phenotypic, ideological, and genotypic heterogeneity. So two-thirds are non-syndromic. It's estimated that about 200 genes are involved in this type of deafness, including more than 100 genes previously described, while 1/3 is syndromic, with more than 400 syndromes associating deafness and other clinical manifestations. Until now, the cause of certain hereditary deafness is still unknown, and many genes are to be discovered. In addition, early diagnosis is very important for effective and rapid management. This is why this study aims to establish the appropriate molecular diagnosis in two consanguineous Moroccan families. So Moroccan families having at least 2 children with hearing loss were recruited to determine the disease causing gene. Then DNA of some affected patients underwent whole exome sequencing. After it was tested negative for the most common mutations involved in deafness in Moroccan families by Sanger sequencing. The whole exome sequencing were performed on HiSeq 2000 Illumina at Autogenetics Corporation in the United States. After that, many bioinformatics tools have been used to analyze exome data and select candidate variations according to filtering approach. Similarly, the molecular modeling approach was used to predict the effect of amino acid substitution on the 3D structure of the protein, and therefore on its functions. So we identified 2 new pathogenic mutations at compound heterozygous state in the GPR98 gene, responsible for Usher syndrome type 2C. For the second family, a new deleterious homozygous PEX1 mutation was identified, causing a disease belonging to the vulgar spectrum disorders. The first family have 3 affected siblings with moderate hearing loss, normal vestibular function, and before installing visual disturbances. The first mutation affects a highly conserved amino acid, and the second mutation leads to a stop codon. Sanger sequencing results confirmed segregation and revealed that the 2 parents and their non-affected child had one of either mutation in the heterozygous state, whereas their affected children had biallelic mutations. The change of proline by threonine was predicted to disturb hydrophobic interactions between two adjacent residues. The second variant leads to stop codon, which results to truncate GPR98 protein, lacking the seven transmembrane and C-terminal domains of the protein. For the second family, two children are affected with profound deafness, impairment vision, and development delay. The two parents are heterozygous, while the two affected patients carried the same homozygous variant. The substitution of the highly conserved amino acid leucine at position 1026 may have led to loss of hydrogen bonds, thus altering the interactions between these residues and valine 1030. Moreover, it's likely to disturb hydrophobic interactions between these residue and its neighboring residue leucine 1032. In conclusion, this is the first time that mutations in the GPR98 gene are described in Moroccan family. And it was only after these two mutations identification in these two genes that the correct diagnosis could finally be established for these two families. And the early genetic diagnosis of these syndromes is of great importance for rapid and adequate therapeutic management, such as bilateral cochlear implantation, favoring the mode of oral communication before the onset of blindness. I would like to thank all the members of the laboratory. And also I thank the families of the patients for their collaboration. And finally, thank you for your attention.
Thank you very much, Dr. Amelie, for your nice presentation. Is there any contribution? Okay. I would like to invite the next presenter, Dr. Kritika Chandra Mohan. The presentation is synergistic anti-cancer activity of terpenoids promotes apoptosis and mitochondrial membrane depolarization in A549 non-small cell lung cancer cells.
A very good evening to one and all present here. Can you hear me?
Yes, we can hear you. Could you do full screen, please?
Yes.
Thank you.
So I'm Kritika, working under the guidance of Dr. K. Pandimadevi ma'am. We are from Alagappa University, Tamil Nadu, India. The topic is synergistic anti-cancer activity of terpenoids, which is promotes apoptosis and mitochondrial membrane depolarization in A549 non-small cell lung cancer cells. So lung cancer is a major threat to compared to all the cancers. This is the most common cause of cancer death in both men and women. But if we take males, this is the major incidence as well as gives mortality to the men. So lung cancer has two types, which is small cell lung cancer and non-small cell lung cancer. This non-small cell lung cancer possesses adenocarcinoma, squamous cell carcinoma, and large cell carcinoma, which is contributed to 85% of the cancers. And the small cell lung cancer, which contributes to 15% of the cancer types. So the treatment includes for the lung cancer, which is chemotherapy, surgery, radiotherapy, and the radiofrequency ablation and immunotherapy. And some of the chemotherapy drugs which is used for the lung cancer treatments are cisplatin, doxorubicin, etoposide, afinitor, and monoclonal antibodies. The drawbacks for the chemotherapy, which induces major side effects like mutation, non-specificity, which also affects the normal cell lines, normal cells in the body, and it limited the bioavailability of the drug, and it induces resistance to the cancer cells, and also it induces the toxicity to the normal cells, and the chemotherapy drug turns the patients into immunocompromised. So we are working on the natural products which will induce the toxicity against only to the cancer cell, not the normal cell. These are some of the natural chemicals which will be using against the lung cancer cells. So here in our study, we have selected the phytol and bisabolol, which is an diterpene alcohol and monocyclic sesquiterpene alcohol, which both belongs to terpenoids. We will use these chemicals, drugs with the combination. Why we are going for the combination? Because and the combination of drugs which will reduce the concentration of the either drug, and it will also induce the multi-targeting potential, and it induce the apoptotic or the cancer anti-proliferation through various modes of molecular pathways, and also it reduces the chemotherapy drug toxicity, and it is safe, and it is approved by FDA. So initially, we have performed the cell viability assay through MTT, and we have found out the inhibitory concentration 50 of phytol and bisabolol, which is phytol is about 25 microgram per ml, and the bisabolol would be 15 microgram per ml. And we have tried the synergistic combination through checkerboard assay. By using the Chow-Tolle method, we found that 12.5 and the 3.75 of the bisabolol and 12.5 of the phytol combination shows in IC50 with the same of the individual component. So further, we have taken the same concentration to elicit the molecular mechanism at which this chemicals induces the apoptosis in the cancer cells. So this is the molecular cellular morphology of the A549 cells. This is a control, and it's a vehicle control. And this is the chemotherapy drug treated cells. And this is the combination of drugs treated cells. This is alone treated cells, phytol alone and the bisabolol alone. We could see the combination of treated drugs shows the membrane blebbing and the apoptotic bodies in the cells. There is a shrinkage in the cell lines. We could see the apoptotic morphologies compared to that of the control. And further, we wanted to see the plasma membrane damage in the cells, and we have performed the LDH and also the propydium iodide staining, which is intercalate into the DNA. And we found that there is a release of lactohydride dehydrogenase, which will reveal that there is a plasma membrane damage. So the intracellular protein get released. And we also see the fluorescence in the positive control and the combination treated cells. We can see the nuclear, which says that there is an plasma membrane damage. The dye is non-permeable. Only the membrane damages, it get into the side cells and intercalate into the nucleus. So further, we want to analyze the apoptotic cells by using acridine origin ethidium bromide staining. We found that the combination of treated cells shows late apoptotic bodies compared to that of the control, and the alone treated sample, which is less inhibition in the cells. So also we further wanted to check the protein levels, the apoptotic proteins. The BAX is an apoptotic protein, BCL2 is an anti-apoptotic protein. We have checked the expression through Western blotting analysis. We found that there is a upregulation in the pro-apoptotic protein and the downregulation of anti-apoptotic protein, which may induce the apoptotics in the A549 cells. And further, we want to measure the mitochondrial membrane integrity and the induction of ROS through rhodamine 1,2,3 staining and DCF-DA. This rhodamine 1,2,3 will go and bind to the mitochondrial membrane present in the cell, and we have checked by addition of the compounds and the combination of compounds. We found that there is a depletion in the fluorescence in the phytol and bisimilar treated cells. So there is a mitochondrial membrane depolarization occurred in the combination of drug treated cells. And also we found that the ROS induction by using DCF-DA staining, we could see the enhanced fluorescence in the combination of drug treated cells compared to that of the control. So from this, we can conclude that there is an ROS induction which may lead to the mitochondrial membrane depolarization in the cell, which will lead to an apoptotic mode of cell death in the A549 cells. Further, we want to check the cell cycle arrest during the drug treatment. We found that through the flow cytometric analysis, we found that the cell cycle arrest was at G0, G1 phase. And further, we wanted to check the toxicity through hemolysis assay. We have checked the combination of drug by using hemolysis assay. We found that there won't be any inhibition in the heme cells. And further, we wanted to check the anti-migration assay by using wound healing method. We found that we have, by using scratch assay, we have performed, and we have given about 24 hours. For the control, it is started closing the gap, but in the treated groups, there is an inhibition. So this drug particularly inhibit the migration of the cells, and thereby this will help in the later stage of lung cancer disease. So to conclude this, we use the phytol and bisimilar for the combination therapy to lung cancer cells, which is A549. We found that it induces intrinsic mode of apoptosis through mitochondrial membrane depolarization, and also it induces the upregulation of BAX and the downregulation of BCL2, which will also induce ROS, which is an oxidative stress dependent action. And this combination of drug causes G0, G1 cell cycle arrest. And it also induces the anti-migration potential of the cancer cells, which will demonstrated to scratch assay. So further, this combination of drugs will be non-toxic to the normal cells, which is RBC and PBMC. So we recommend this drug could be a possible candidate for treating non-small cell lung cancer disease. Thank you.
Thank you very much for your nice presentation. I think we have solved the 8 presenters' technical problem. So I would like to announce Dr. Salatin Redon again. The title was In Silico Screening and Exploration into Phenotypic Alterations of Deleterious Non-Synonymous Single Nucleotide Polymorphisms.
Thank you, Doctor. Do you hear me right now?
Yes, we can hear you.
Great, thank you. So I'm going to share my screen. Can you see my full screen?
No, yes, now we can see. It's okay.
Okay, great. Hello, everyone. My name is Salah Derdoan, a PhD in my second year of research. I'm working on genetics and genomics of male infertility and disorders of sex developments in the Moroccan population. So in this work, me and my team did an in silico screening and exploration into phenotypic alteration of deleterious non-synonymous single nucleotide polymorphisms in the CYP11A1 gene which could be a potential genetic factor to predisposition in the disorders of sex development. So the CYP11A1 gene is a gene located in the chromosome 15 in the long arm Q in the region 2, band 4, sub-band 1. This gene codes for cholesterol side chain cleavage enzyme, or simply called by cytochrome P415 SCC. So this cytochrome belongs to the superfamily of cytochromes P415, which are hemoproteins. They carry a heme molecule as a cofactor, and they intervene as oxidoreduction catalyzers for large and small molecules. There are 57 types of them encoded by human genome, and they are classified dependent on their intracellular location. 7 of them are classified as mitochondrial, and 5 between these 7 are implicated in the steroidogenesis. They are mainly expressed in the adrenal glands on top of the kidneys. And the cytochrome P415 SCC, which is coded which is coded by the gene of our interest, is one of them. So the P415 SCC cytochrome catalyzed the first step of lateral chain cleavage in the which transformed the cholesterol molecule to pregnenolone and isocaproic acid. So this cleavage is a 3-step transformation, which is characterized by 2 metabolic intermediates, 22-hydroxycholesterol and 20-22 dehydroxycholesterol. So according to many research, the first step is a limiting step in the steroidogenesis, which make from the P415 SCC cytochrome a limiting and crucial enzyme in the steroid hormone synthesis in the human organism. So this cytochrome can also be considered as a cofactor, or can be considered as an enzyme which could be implicated in many adrenal insufficiencies. One of main adrenal insufficiencies is disorders of sex development, especially for males. So in order for a complete reproductive system for male development, there are 2 important genetic triggers, which are genetics and hormonals. So the hormonal trigger is characterized by testosterone secretion during the 8th week of human male embryo's development. When the steroidogenesis is not stable, or if there is an insufficiency in this area, the testosterone secretion will not occur, which could lead to a disorder of sex development. So it seems like the CYP11A1 gene is an important gene which could be conserved and not exposed to mutations and deleterious genetic variations like copy number variants or SNPs. So SNPs, or simple nucleotide polymorphisms, are the most frequent genetic variations, and they could be implicated in many pathologies because they could predispose many subjects of many health problems. And SNPs are mainly located in non-coding regions, but there are lots of them in the coding regions. The most easy one to study are non-synonymous or missense SNPs, because they are susceptible to cause structural and functional modifications on the encoded proteins. So the aim of this study is to define the most deleterious non-synonymous SNPs of the CYP11A1 gene, which could lead to a predisposition to an insufficiency of steroid hormone synthesis. So there are many non-synonymous SNPs of the CYP gene in the DBSNP database. A systematic step-by-step elimination is essential to identify the most deleterious one between them. So as a first step, we retrieve 8,917 non-redundant CYP11A1 gene SNPs from the DBSNP, and only 389, which correspond to 4.33% of the total, were classified as missense or non-synonymous between all of them. So we choose them and we eliminate all the others. After that, we did a prediction or an effect prediction by using 4-chain prediction algorithms. Those algorithms are based on many bioinformatical and empirical data to predict the effect of each substitutions caused by each SNP on the encoded protein. So according to 4-chain prediction algorithms, only 11 non-synonymous SNPs out of the total of 389 were predicted having a deleterious effect on the P415 SCC cytochrome. So we select them and we systematically eliminate all the others. After that, we did a conservation analysis of the P415 SCC cytochrome protein sequence by using the Conserve web server. According to Conserve results, only 9 non-synonymous SNPs out of 11, which we selected previously, correspond to 8 substitutions located on highly conserved and exposed and potentially functional positions, which means that those 9 SNPs could lead to modifications in positions that should not be modified in a protein. So we choose them as the most deleterious, and the rest of the 11 were systematically eliminated. And after that, we retrieved a 3-dimensional structure of the P415 cytochrome protein from the protein data bank. So in order to introduce each substitution in this structure, so systematically we had 9 different copies of this 3-dimensional structure. Each one carry a substitution which correspond to one of the 9 non-synonymous SNPs and the most deleterious one between them. And we also had a wild type one which gonna serve as a control. After that, we did an energy minimization in order to adapt each variant conformation to its lowest energy possible stage. And this procedure was done using Yasara web server. So after this minimization, we received all the results and we did what we call stability assessment by using DH consensus server, which predicts the effect of each substitution on the overall protein stability. So according to all 3 algorithms applied by DH server, only 6 substitutions out of 9 were destabilizing to the P415 cytochrome protein structure. So we kept them and we eliminate the rest of the 9. In this stage, we were ready to do a protein ligand docking and protein ligand affinity assessment. So we used AutoDocVina software to dock the cholesterol in the 6 variants of the P415 SCC cytochrome for measuring the affinity of each interaction. The affinity corresponds to the binding energy between the ligand and the protein. So substitutions causing a low affinity interaction have binding energy greater than the wild type one, which correspond to minus 10.1 kilocalorie per mole. So according to AutoDocVina results, all the 6 substitutions have binding energy greater than the wild type one. But the most destabilizing one was the substitution where in the position 138, where glycine was replaced by arginine, with a binding energy equal to minus 7.9 kilocalorie per mole. So as a conclusion, these findings show interesting genotype-phenotype correlation between deleterious non-synonymous SNPs and non-stable cytochrome cholesterol interaction. These non-synonymous SNPs could predispose male subjects to adrenal insufficiencies, health problems, including disorders of sex developments. Further, in silico, in vitro, and in vivo tests should be done for more accuracy. And as a goal for this, as a continuation for this study, we are aiming to do molecular dynamic simulations for these 6 substitutions and assessing the molecular kinetics of ligand protein complexes. Thank you for your attention.
Thank you very much for your nice presentation. From the authors, is there any contribution? Thank you very much. Now, the 11th presentation Dr. Adila Valiyeva from Azerbaijan, chemical investigation and antimicrobial activity of some Hyoscyamus species from Azerbaijan.
Have a nice evening. Thank you very much for chance and such a nice opportunity. My topic is chemical investigation and antimicrobial activity of some Hyoscyamus species from Azerbaijan. Two Hyoscyamus species we investigated in our research. Both Hyoscyamus species are well-known source of tropon alkaloids, and such as atropine, hyoscyamin, and scopolamine. In traditional medicine, hyoscyamus species were used as in the different diseases like asthma, spasmodic cough, convulsions, neuralgia, and etc. Different group of compounds we investigated in our research. Alkaloids are the main compounds of this species, of course. Phenolics, total flavonoids, and phenolic content, fatty acid, phytosterols, tyramine derivatives, and alkanes, saturated hydrocarbons. Hyoscyamus liver alkaloids were determined by using first the GCMS method. Approximately 30 alkaloids, mainly tropon, non-tropon, and pyrrolidine derivatives identified by using GCMS method. First, 4 alkaloids are the major alkaloids, of course, hyoscyamin, napatropine, naproskopolamin, and scopolamine, tropine, alpha-beta tropine, and epoxytropon, also nortropon and pyrrolidine derivative. You can see in this identification, and they are also other main compounds of the plant. Hyoscyamus reticulata alkaloids are also firstly identified by using GCMS method. Hyoscyamin, scopolamine are the major compound, and phenylacetoxytropon, hydrohyoscyamin, nortropine, also higrin, two different higrin derivatives were observed in the different organs of Hyoscyamus reticulatus. LCMS determination also was used to identify some of alkaloids. 7-beta-hydroxyhyoscyamin, also hyoscyamin N oxide, norhyoscyamin, anti-gloyloxytropon, 1-miltropine were identified by using LCMS fragmentation. You can see here identification of 7-beta-hydroxyhyoscyamin. Quantitative analysis of atropine was carried out by using HPLC method at 109 nanometer in comparison with analytical standard of atropine sulfate, and amount of atropine in the Hyoscyamus species were presented here. 0.12 milligram per gram of the Hyoscyamus reticulatus and 0.22 milligram per gram of the Hyoscyamus niger sample were identified. And tyramine derivatives, NCs and intransferroid tyramine derivatives were identified by using LCMS fragmentation. Their molecular ion and characteristic fragment ions helped to identification. Rutin was determined as a major flavonoid of the plant Hyoscyamus leaf, both Hyoscyamus leaves, and you can see here the major peak of the rutin and other phenolic and flavonoid compounds of Hyoscyamus reticulatus leaf here. Camphorol hexosic deoxyribonucleic acid also is a flavonoid from the both Hyoscyamus species, mainly their leaves, and their camphorol part and their molecular ion. Its molecular ion helps to identification. For the fatty acids, main fatty acids of both species, both seed oils of plants are the linoleic and oleic acids, which are unsaturated fatty acids and has importance in the medicine and cosmetic and other fields. Palmitic and stearic acid also were identified in both species, but not in the higher amount, approximately 2 and 4% in the samples. Phytosterols were qualitatively determined by using GC feed method. 3 phytosterols were determined: campestrin, stigmasterin, and beta-sitosterin in both seed oils of plants. For the Hyostium niger fruit, we can observe in their hexanoic and chloroformic extract, we identified alkanes, different alkanes as a meiopeaks. Tetracontan, tetracontan, and monohexacontan are the main compounds of this hexanoic and chloroformic extract. CCMS was used for the determination. Total amount of flavonoids and phenolic compounds was determined by using spectrophotometric method. Microgram per gram equivalent were present in these slides. Different picrohydrazine activity were determined as the same way by using spectrophotometric method. Microgram Trolox equivalent per gram dry plant sample were present in this table. Antimicrobial activity of plant leaf and seed extracts tested against different microorganisms, mainly Staphylococcus aureus, Bacillus cereus, Salmonella enterica, Listeria monocytogenes, and Escherichia coli. Significant activity was used in the concentrated sample, in the higher concentration, I can say. For the all antimicrobial analysis tests, we can see different results, different zones of inhibition were presented. Depends on the results. Thank you for your attention.
Thank you, Dr. Velieva, for your nice presentation. And the next presentation is, I think, the genetic researchers in Morocco is very hardworking. And the next 12th one is from Morocco, Dr. Imane, for the presentation.
Hello, can you hear me?
Yes.
Can you hear me? Hello.
Yes, we can hear you. Hello.
I will share my my screen.
Mm-hmm.
Can you see the screen?
Yes, could you do it full screen, please?
Okay, great.
Thank you.
Hello, everyone. I would like to thank you for this opportunity. I'm Aitarais Iman, PhD student at Pasteur Institute of Morocco. I will present to you my study, Genetic Heterogeneity in GGB2, code for A3, ATP6V1B1, and the EDNRB variants detected among hidden impaired families in Morocco. So firstly, deafness is one of the most frequent neurosensory disease. It affects 1 at 2 per 1,000 newborn. Hearing loss have heterogeneous causes, genetics and environmental. It can be either isolated, non-syndromic, or associated with damage to other organs, syndromic. In Morocco, the GGB2 gene is the main cause of hereditary non-syndromic hearing loss. The most prominent variant in Morocco and the Mediterranean region is the deletion 35-del-G. The second cause of deafness in Morocco is the presence of the substitution 242G4A located on LR80ONT gene. Among the syndromic genes linked to deafness and which occur in the Moroccan population, MIO7A and PEX1. For non-syndromic genes, there is GMT1 and PGVK. In this study, all exome sequencing analysis was used to resolve this etiology of hearing loss in 6 Moroccan families. For patient and methods, for patient recruitments, we recruited 6 consanguineous families with syndromic and non-syndromic hereditary deafness. DNA extraction was done by QIAGEN kit. The whole exome sequencing relies on library preparation, cluster generation, sequencing, and data analysis. To determine the segregation with the phenotype of the disease in these families, Sanger sequencing was carried out to validate the mutation in the candidate gene. Finally, the bioinformatic analysis, proteins and interactions analysis and visualizations were performed using Yasara software. Results for the 6 families analyzed, results were obtained just for 4 families, as we see in the table. The whole exome sequencing results revealed a novel homozygous mutation of the EDNRB gene, which has been predicted to be possibly damaging using CIFS-FATMAN and polyphen programs, deleterious by LRT software, and disease causing by mutation tester. Heterozygous variants already described in GGB2 gene involved in a dominant form and predicted to be damaging by CIFT, polyphen, and FATMAN, disease causing and deleterious by mutation tester and allergy. Homozygous variants in COL4A3 gene predicted to be damaging, disease-causing, and deleterious by these prediction tools. Finally, a homozygous insertion variant of ATP6V1B1 has been identified in the patient, which it's predicted to be damaging and disease-causing too. For the mutation found on EGA and RB gene, we studied the effects of the mutation on the stability of the 3D structure of the EGA and RB. And as we see in the table, a destabilizing effect on the EGA and RB 3D structure was predicted based on the 4 different tools. The potential structural impact of the mutation of the EGA and RB gene is revealed by molecular modeling and analysis of amino acid conservation. As we see, yellow dot lines represent hydrogen bonds and green lines represent hydrophobic interactions. The amino acid interactions analyzed showed that this variant did not affect hydrogen bonds. However, it might disturb the hydrophobic interactions between the residue in 409 position and its adjacent amino acid, as we observed that the interaction with leucine 405 residue was replaced by one with glycine 410. For discussion, in this study, we have described 4 genes among them, which are described for the first time in the Moroccan populations, and other already described. GGB2 variants was observed for the first time in the Moroccan populations, but it was previously reported in many studies, such as Ghana, China, and India. COL4A3 mutations observed for the first time in the Moroccan population, but it was previously described in 3 families in Italy. ATP6V1B1 variant was previously observed in many studies, Mexican, Moroccan, and Tunisian populations. And finally, the novel EGA and RB mutations was linked to Waardenburg syndrome type 4, whereas other studies suggest that EGA and RB should be considered as another pathogenic gene prevalent in Waardenburg syndrome type 1 to the heterozygote states. For conclusion, the study described the involvement of genetic variants of the gene GGB2, COL4A3, EGA and RB, and ATP6V1B1 gene in syndromic and non-syndromic deafness in Moroccan patients. The results can be considered very important because they allow to show even more the genetic diversity of hereditary deafness in Moroccan patients. Thank you for your attention.
Thank you very much, Dr. Imane, for your nice presentation. Now I would like to invite Dr. Harmak for the presentation about male infertility. Yes, Dr. Harmak. Okay, I think the doctor is not here. Okay, I will pass to the next presentation. I would like to invite Dr. Sabika Frasat about the presentation called Analysis of Demographic Characteristics and Risk Factors of Breast Cancer in Southern Punjab, Pakistan.
Hello, everyone. Can you hear me?
Yes, we can hear you. Could you please do full screen?
Yeah. Okay, thank you. Bismillah Rahman Rahim. I am Dr. Sabika Farasat. I am working as Assistant Professor at Department of Zoology, Faculty of Biological Sciences, Qaeda University, Islamabad, Pakistan. And the topic of my today's talk is analysis of demographic characteristics and risk factors of breast cancer in Southern Punjab, Pakistan. So here is the brief introduction of this work, which I'm presenting on behalf of my co-author as well. So globally, breast cancer is actually a very devastating disease for females, especially, and 1.7 million new breast cancer cases are diagnosed each year out of them 33% die. In Pakistan, this malignancy is 2.5 times higher than the neighbors India and Iran, and that is very unfortunate. And mostly another unfortunate thing is that in Pakistan the females are diagnosed at advanced stage of the disease, and therefore mortality rate is also comparatively high as compared to the advanced countries. If we look at the risk factors that how this malignancy occur and that why and which are the contributors to this cancer, and there are about 10% cancer which have hereditary background, that is they are genetic, and the other multiple risk factors are also reported for breast cancer, which are related to the lifestyle factors and sometimes ethnicity. For example, white women are more prone to breast cancer as compared to the Asians, but in Asians it is reported that the disease is more vigorous as compared to the white woman. And the reason can be that in developed countries there is early screening and early diagnosis and disease can be controlled, but in case of underdeveloped country like Pakistan, a developing country like Pakistan, so there is late recognition of the disease, and therefore it is there at advanced stage, and there are more chances of death. So among reproductive risk factors, age at menarche, age at first full-time pregnancy, age at menopause, parity, breastfeeding per child, history of miscarriage, use of oral contraceptives and hormone replacement therapy are actually considered as contributors to the breast cancer. So there are multiple reports from various areas of the world, different countries, but in case of developing country like Pakistan, which are the major risk factors are contributed, there is very less information regarding that. And especially the population of South Punjab region of Pakistan, it is very less explored with regard, and it is not explored with regard to the risk factors of breast cancer, and therefore there is a need to work in this area. So the aim of this study was to investigate epidemiology, clinical aspects, and risk factors associated with breast cancer in Pakistani southern Punjab region. And for that purpose, a case control study was designed, and which was conducted from October 2017 to March 2018 for 6 months. And ethical approval for this study was taken from Bioethical Committee of Qaeda University, Islamabad, and samples were collected from Bahawalpur Institute of Nuclear Medicine and Oncology, that is BINO, and Bahawal Victoria Hospital. So the sample size we calculated by using EPI2 software, taking 5% margin of error, hazard ratio 2, and with power 80%. And all those females which were actually diagnosed by biopsy samples, they were included in the study, and only those females which have any other disease, any other cancer other than the breast cancer, or which were not willing to participate in the study, they were excluded from the study. So this because it was a case control study, we also enrolled healthy control samples in the same time, and all the patients and the controls, they signed the informed consent for participation in this study. So we designed a questionnaire using the previous reported literature, and these questionnaires were filled, which included the information on socio-demographic, menstrual, and reproductive characteristics, family history of cancers in first and second degree of relatives, and several lifestyle factors, including night shift working, smoking, etc. And we also noted the height and weight of the patients and controls to calculate the BMI, and the physical activity was also estimated in 3 subsets: activity at work, activity of traveling, recreational activity, as well as sedentary behavior. And all these were recorded using the Global Physical Activity Questionnaire, and which is developed by WHO, and to estimate the physical activity of the individuals. After collecting all the data was recorded in the Excel files, and SPSS and Stata were used to analyze the data. Qualitative variables were expressed as frequencies and percentages, while continuous variables were expressed as mean standard deviation. So we performed conditional logistic regression model to identify potential risk factor linked to breast cancer risk in the southern Punjab population, and in our sample which we collected, and crude and adjusted odd ratios obtained were used to estimate contribution of each factor for all odd ratios, 95% confidence interval were calculated. So bivariable, that is unadjusted, and multivariable analysis were also conducted, and we only after calculating all the risk factor, we statistically significant results were actually considered as the main contributors. So in our results, as the study was cases and control, and all the cases and controls were age matched by age relaxation within 1 month period. Out of there were total 326 women who participated in this study, and out of them 163 were cases, and same number was of controls. So we after data analysis, we found that positive family history and hypertension, they are significantly linked to increased breast cancer risk in our study population. And the two main things, family history of cancer and hypertension. So intense physical activity, increased anthropometric measurements, and breastfeeding per child in months. If it is more, they were inversely associated with breast cancer. It means these are actually not contributing to, or these can be considered safe, or they were negatively linked to breast cancer risks. So every outcome is mentioned in each table, and here are some tables. The table 1 shows the data of continuous variable presented as mean standard deviation, and you can see that age in cases and control, as both were age matched, so it's same. Age at menarche, it's a bit different, higher in cases as compared to control. Age at menopause, BMI ratio, age at first full-term pregnancy, and breastfeeding per child. And here you can see that breastfeeding per child is less in case of cases as compared to controls, and number of live births was also calculated. So here are the clinical characteristics of breast cancer patients, which shows that most of the patients, they were of a unilateral breast lump, and type of carcinoma, in most cases invasive ductal carcinoma was found. That was in 142 patients, and we also found recorded the estrogen receptor, progesterone receptor, and HER2 NEU status for all the cases. And the site of breast affected were also noted, and in mostly there was unilateral. In 65 cases it was the right breast, and in 92 it was the left one, and bilateral was only in 6 cases. And size of breast lump was also checked. That was in 92 cases it was 6 to 8 centimeter, and metastasis to lymph node was also found, and it was found that axillary lymph node cancer metastasized to axillary lymph node in 121 cases, and to axillary supra clavicular, opposite axillary, and no lymph node involved are not examined. Stage of tumor, and you can see that there was no patient at stage 0, and there were 50 patients at stage 4, but stage unidentified was for 68 patients, in which it was not recorded at the time of enrollment for the study. So among the reproductive risk factor, age at menarche, breastfeeding per child, history of miscarriage, parity, and age at first full-term pregnancy all were recorded, and it was found that age breastfeeding per child was a protective factor, because that if there is more breastfeeding per child, then there is less risk of cancer in our study population. Medically related risk factor were hypertension and family history, and both were positively linked to the risk of breast cancer. And among lifestyle factor, use of some what you can say tobacco and physical activity were also recorded. So out of 163 patients, 53 were aged 26 to 40 years, 84 were aged 41 to 60 years, and 15 were aged 61 to 80 years. And average age at diagnosis vary among different populations, with mostly reported at 60 years. But in our current study, it was 46 years. And it means that there is early onset as compared to the other reports. So none of 163 patients were at stage 1, and 19 were at stage 2, 26 were at stage 3, and 50 were at stage 4. And that is considered that it is a very alarming, because most cases are diagnosed at later stage, and therefore they do not respond to therapies, and there are more chances of death of the patient. So 5 factors remain significantly associated with the breast cancer, including breastfeeding duration per child, hypertension, family history, physical activity, and BMI. So main was hypertension and family history, but the physical activity and BMI, they were also considered that they were higher, they were linked to the increased cancer risk, and that was also reported in other previous studies. There was positive association of hypertension family history with increased breast cancer risk, while breastfeeding, increased BMI, and intense physical activity decreased the risk in one from southern Punjab population. However, increased BMI is considered as the risk for breast cancer, but here this increased BMI was related to less risk. The reason was that increased BMI was also found in our control cases. So there was no difference in the data of cases and controls. Therefore, we found this disputing result. Lifestyle and reproductive risk factors may take many years before their contribution to cancer incidence is noticed. So the preventive measures and awareness campaigns are indispensable to reduce breast malignancy. So this data is also published in this Pakistan Journal of Medical Association. So the reference is given here. Thank you all for listening me.
Thank you very much, Dr. Prasad. Now I would like to invite Dr. Hado Harmak for their presentation about male infertility. Dr. Hado Harmak.
Hello, can you hear me?
Yes, yes, we can hear you.
So I will share my screen. Hello, everyone, and welcome to my presentation. First of all, I would like to quickly introduce myself. I am Harmak Houda, a PhD student from Genomic and Human Genetic Laboratory at Pasteur Institute of Morocco. And today, I'll be presenting the results of my article about the computational analysis of the potential impact of MTC complex missense SNPs associated with infertility. During meiotic prophase 1, chromosomes undergo rapid movement that is the formation of inter-homolog recombination intermediates and the line synapses crossing over and segregation process and ensure the pairing and eventually recombination of homologous chromosomes. The recently discovered triplet complex made of telomere repeats binding pocket formation protein 1 and 2, TERB1 and TERB2, and monomer anchor junction protein MAGEN has been demonstrated to be involved in this process by tethering telomere ends to the nuclear envelope, transmitting cytoskeletal force via the link complex to drive this rapid movement and assembling of meiotic mammalian telomeres of the nuclear envelope of telomere. Disruption or mutation of any of the MTC genes, TERB1, TERB2, or MAGEN, cause meiotic arrest with loss of telomere attachment and chromosomal movement, failure of DNA double-strand break repair, and disrupted synapses. Accordingly, the disturbance of telomere nuclear envelope connection impairs the homologous insertion that cause meiosis defects and disordered synapses, leading to infertility. The study aims to identify the deleterious SNPs in the TERB1, TERB2, and MAGEN genes by predicting the significant pathogenic impacts on the function, stability, and structure of TERB1, TERB2 complex and TERB2 MAGEN complex using bioinformatic tools as a systematic in silico approach. Figure 2 shows the schematic representation of in silico workflow used in this study. Figure 3 shows the 3D dimensional structure of complex A, TERB1 and TERB2 complex, B, TERB2 and MAGEN complex. In this study, figure 4 shows the mutation classification and consequence type of TERB1, TERB2, and MAGEN genes. In this study, only missense mutation that could have been mapped on the protein structure were included, which are 18 variants of TERB1, 31 variants of TERB2, and 21 variants of MAGEN. Out of 70 missense SNPs, only 30 were the series using the VIP tools such as SIFT, Polyfen, Mutation Tester, MetaIR, and MetaBind Web Server. They were the series with a change in binding free energy superior or equal to 1.5. Figure 5 shows the heat map of global Biden energy score of TERB1, TERB2 complex and TERB2 Majan complex. Region with the lightest orange shade represents complexes with higher free band energy, which correlates with lower Biden affinity. This region are represented by 6 mutated complex of TERB1, TERB2 and 2 complex of TERB2 Majan. The protein stability was further evaluated by Duet web server. The results of MCDM and Duet algorithm showed that 26 amino acids were recorded at destabilizing. Conserve web server was used to identify the conserved domain in the TERB1, TERB2, and Majan protein sequence. The mutated structure were further analyzed by Yasara view with regard to evaluate confirmation of variation by calculating their MSD. 31 mutation out of 32 showed higher MSD value, which indicates thus 4 variants that reflected maximal structural dissimilarity were used for superimposing. Reside deep web server is applied to predict the effect of SNPs on post-translational modification on the human TERB1, TERB2, and Majan. Reside deep identified sites for all-linked glycosylation for TERB1 and TERB2 protein and S-palmotylation for TERB2 protein, and no PTM site has been located on Majan protein sequence. Furthermore, a cumulative score was calculated for the 32 damaging variant based on 7 in silico. All of the listed 32 SNPs were predicted as deleterious or damaging by SIFT, polyphen 2, and metabine. Only 13 amino acids were considered as highly pathogenic SNPs since they were agreed significantly by the Purdue tools with a score of Biden affinity superior or equal to -11. 12 variants revealed negative unfolded free energy predicted by the tree algorithm, which indicates that they stabilized the protein stability, except for one residue that was destabilized only by 2 algorithms. Among the 13 mutations, 12 showed higher MSD value, except for one mutation that had a MSD value equal to 0. The result of the concept analysis showed that the 13 amino acid change were located in conserved region with a score of conservation of 9. So in discussion, in the current study, we used variant algorithm. So we can identify the most deleterious SNP of the TERB1, TERB2, and Majan genes. Accordingly, 13 SNPs were cumulatively predicted to be pathogenic by at least 6 algorithms, decreased the stability, and were located in conserved region, which may affect the structure and function of protein. Bioinformatic analysis provided powerful insight into understanding and predicting the effects of mutation on protein structure, including how mutation alter protein stability, binding energy, binding affinity, evolutionary conservation, and interaction with other protein and the relation to the severity of genetic disease. So in conclusion, in our work, out of 32 missense mutation, 13 were cumulatively predicted to be high-risk pathogenic SNPs. Combination of multiple in silico tools provided information required to predict the effects of this mutation on the functional and structural characterization of TERB1, TERB2, and Majan protein. Our finding added to previous knowledge and supported earlier studies that might be helpful for further understanding the role of meiotic telomere complex in mal-infertility. Nevertheless, bioinformatics tool cannot service conclusive experiments, and the results should be verified by supplementary data. And thank you for your attention.
Yes, thank you very much for your fast presentation. It was nice, a nice research. Thank you, doctor. And the next presentation belongs to Rutaba Gül. Neurological manifestations in Pakistani lysosomal storage disorders patients and molecular characterization of Gaucher disease.
Hi, everyone. Can you hear me?
Yes, we can hear you.
Okay. First of all, I would like to thank to Congress for providing such an amazing opportunity. And my name is Rutaba Gül. I'm a PhD scholar at Department of Zoology, Quaid-e-Azam University, Islamabad, Pakistan. Today, I'm going to present one of my recent publications entitled as Neurological Manifestation in Pakistani Lysosomal Storage Disorders, Patient and Molecular Characterization of Gaucher disease. In introduction, the lysosome is a cell organelle to regulate the function and degradation of cellular macromolecules. The substrate for lysosomes are obtained through multiple pathways like phagocytosis, autophagy, and endocytosis, and further they are degraded by lysosomal hydrolases. If this function of lysosomes got disturbed due to absence of any enzyme, it may lead to lysosomal storage disorders called LSDs. LSD is a large group of inherited metabolic disorders with 2/3, including the central nervous system involvement. Most of the LSDs are inherited as autosomal recessive disorder, except Fabry and Hunter. These are inherited as X-linked diseases. Frequency of LSDs in different population is 1 in 7,500 live births. However, exact prevalence in some population like Pakistan is even higher due to high rate of consanguinity among the population. The treatment options include enzyme replacement therapy, pharmacological chaperone therapy, bone marrow therapy, and human stem cell transplant. These are the frequently used treatment options. There are some other options, but they are in their clinical trials. The neurological manifestations are present in some forms of LSDs, like Gaucher, Niemann-Pick disease, and in few mucopolysaccharidosis cases, ranging from mild to severe, and in early ages to adult years of life. The aim of current study was to report the neurological manifestations in Pakistani LSD affected cases and to explore the genetics of Gaucher disease affected patients. In methodology, firstly, the study was approved by Bioethical Committee of Quaid-e-Azam University, and further we enrolled 45 cases from Children's Hospital, Pakistan Institute of Medical Sciences from January 2018 to December 2019. Diagnosis was made completely on the basis of their clinical profiles at the time of presentation. Family history was taken, the pedigree was drawn, and the blood samples of the Gaucher patient and their accompanying individuals were collected after signing the consent from them to participate in the study. In results, among adult families, 28 out of 45 families had neurological manifestations. However, the majority of patients were from Punjab. 31 out of 45 families were from Punjab region of Pakistan, while 10 families were from Khyber Pakhtunkhwa and 4 families from Azad Jammu and Kashmir state. 43 out of 45 families have had first cousin marriages. Here you can see in the ethnicity, 3 families were from Punjab, while 2 families, family B and E, were from KPK, the Khyber Pakhtunkhwa region, while the parental consanguinity is present in all of the 5 families, and neurological manifestations were present in 4 families out of 5. But in family A, there was no neuronal manifestation along with other clinical features. These are the pedigrees, and you can see there are first-degree consanguinity among all 5 families. In this table, I would like to focus on the enzyme activity that was checked in 4 families out of 5, and you can see in 4 families, the enzyme activity GKs was lower than the cutoff values, and in genotype, the DNA mutation and the protein change, and you can see that we have the homozygous state in L483P form in protein. This is the chromatogram showing the homozygous mutant, the heterozygous carrier, and the homozygous wild type, while the last D figure is showing the amino acid substitution. Green one is showing the wild type, while the red one is the mutant one. 45 cases, there are 45% of cases of neurological manifestations due to LSTs. The Mendelian recessive inheritance of LSTs in Pakistan is more common due to consanguinity. The most prevalent subtype is MPS, mucopolysaccharidosis, that was 35 out of 45 cases. The previous reports were also published from Pakistan, including the MPS subtype 1. Here you can see one of the nonsense and one missense mutation already published by our group. MPS 2, 3, and Niemann-Pick, Gaucher, and Griscelli diseases have also shown the neuronal involvement. Gaucher disease has 3 subtypes. The first one with no neuronal involvement, while 2 and 3 with neuronal manifestations. In GD patient, neurological manifestation was shown by all 4 families except family A. The identified variant L483P, according to literature, is responsible for causing all 3 subtypes of GD. And our findings suggest such a high prevalence of this variant in our population. In family A, the neuronal involvement is although not present at the start, but the possibility at later stage of life cannot be excluded. This variation affects the catalytic domain of enzyme beta-glucosidase, that this causes the loss of enzyme function. 62% of neuronal manifestation in LSD cases is response in our findings that highlights the dire need of study to explore Pakistani population molecular basis for LSD. Identification of alpha-83P in all families is a high frequency prevalent variant to be further explored. I would like to thank all the participating family in this study, staff of Children's Hospital, PIMS Islamabad, and our colleagues for helping one way or the other. Thank you so much.
Thank you very much. I'm wondering about Islamabad. And the next presenter is Dr. Rehan Khan. They are going to present synthetically lethal nanoparticles for the treatment of colorectal cancer by targeting cancer cell-specific vulnerability.
Yeah, good evening, everyone. Can you hear me?
Yes, we can hear you.
I'm sharing my screen now.
Mm-hmm.
Can you see my slide?
Yes, it's okay. Thank you.
Yeah, so my name is Dr. Rehan Khan, and actually I'm working in an institute on like dedicated institute in India, which are like mainly working on the developing nanoparticle for the addressing the health in various diseases. So the topic of my today's talk is synthetically lethal nanoparticle for the treatment of for the treatment of colorectal cancer by targeting cancer cell-specific vulnerability. And the colorectal cancer is the fourth most common cause of cancer worldwide, and over 1 million cases diagnosed worldwide each year, and the fifth most common cause of cancer-related mortality in Indian men and women. And the concept of synthetic lethality, there are like various types of treatment regimen are already available in the clinic, but there's a newer concept. The concept is synthetic lethality. Synthetic lethality is a concept in which we are exploiting the cancer cell-specific somatic mutation or genetic alteration. So we are trying to explore the partner of the this particular mutated gene, and we are trying to suppress, inhibit, or silence the synthetic lethal partner of that particular gene to specifically kill the cancer cells, because that particular mutation will not be present in the normal cells. So there will be like very less collateral damage on the normal cell. So the basic definition of the synthetic lethality is a rare genetic interaction between two genes which are not lethal on their own, but when present together, it lead to the cell death. So as you can see here, the example is simple. Like suppose there are two genes, gene A and gene B. If gene A is mutated in a cell and the gene B is wild type, the cell will survive. And in the opposite manner, the gene A is wild type and gene B is mutated, then still the cell will survive. If both gene A and gene B both get mutated, or then it will lead to the lethality. It means like there is a interaction, synthetic lethal interaction between gene A and gene B. In the cancer context, we are exploiting this particular concept in the to develop a nanomedicine which will suppress the one synthetic lethal partner of that particular mutated gene. So in this study, we are particularly using the Bloom syndrome. The Bloom syndrome is reported to be somatically altered in colorectal cancer and in various other type of cancers. And it is an evolutionarily conserved tumor suppressor gene, and it plays a role in the homologous recombination repair of the DNA. And it's already reported that there's a synthetic lethal interaction between Bloom and SOD1 enzyme, superoxide dismutase 1 enzyme. So there's about the SOD1, is an evolutionarily conserved synthetic lethal interactor of Bloom gene. And it produces the ROS, and it will lead to the generation of ROS is responsible for that inducing DNA double-strand breaks, and SOD1, if you inhibit the SOD1, it will lead to the excessive ROS removal, and it is like play an important role in the detoxification pathway in cells. Because SOD1 is responsible for the conversion of superoxide anion radicals into the H2O2, and then for the PRDX2 enzyme act on this to convert this H2O2 into water. It is a detoxification pathway, and it is highly conserved irrespective of the like from a simpler organism to the complex organism. And the LCS1 is the drug which is the specific inhibitor of the SOD1 enzyme. But the problem is LCS1 drug has a very low solubility, aqueous solubility. So the clinical application of this drug is a challenge. So to overcome the limitation of that particular drug, we have developed a nano formulation. That is, we develop an iron oxide nanoparticle. The advantages of the iron oxide nanoparticle, it can be used for the magnetic hyperthermia, and we can also like load the drug inside this nanoparticle, and we can magnetically drive this particular nanoparticle to the site of the tumor. But the problem is this particular magnetic nanoparticle is reported to induce ROS generation, and it may cause toxic effects. So to overcome the toxic effect of this nanoparticle, we coated this magnetic nanoparticle. As you can see here, the magnetic nanoparticle is the core. We coated this magnetic nanoparticle with three polymer layering. So first we coat the aminocellulose layer to reduce the toxicity and to further functionalize the biocompatible and biodegradable layer 2, that is the PAMAM dendrimer. And in the like, as you can see here, that in the pink region, that like this is the PAMAM dendrimer, and we conjugated the PAMAM dendrimer to the aminocellulose layer. And this PAMAM dendrimer creates the pocket for the encapsulation of the drug. You can see here that the structure of the LCS1. LCS1, like it creates the pockets in like so that the drug can encapsulate inside this nanocarrier. And at the periphery of this nanocarrier, we conjugated the PEG. Polyethylene glycol. Polyethylene glycol is like reported to induce hydrophilicity as well as to prevent the particle aggregation. And if we inject the particular nanocarrier into the circulation, into the then it like enhance the circulation resistance time also. So therefore, we have customized this particular nanocarrier for the encapsulation of this LCS1 drug. So this is the schematic representation, how like this superoxide anion radical generated by the cells, like and SOD1 enzyme is convert superoxide anion radicals into H2O2, and then PRDX2 enzyme converts this H2O2 into water. And if we inhibit this SOD1 enzyme, and this superoxide, both superoxide anion radicals and H2O2 are responsible for causing DNA damage, specifically DNA double-strand breaks. And to repair DNA double-strand breaks, there are two particular pathways. The main pathway is homologous recombination repair pathway, which is an error-proof pathway. And cells will try to get like a repair their DNA damage through this pathway, HRR pathway. But due to the deficiency of bloom in the cells, this pathway will be defective, but still cell will try to adopt the alternative repair pathway, that is non-homologous end joining, and then it will lead to the inefficient DNA repair, and it will lead to the lethality. So this is the specific mechanism, like how like this, if we target the SOD1 enzyme, then how like bloom defective cancer cells will be killed, and while bloom wild type cell will be least affected. So this is the schematic representation of like stepwise synthesis of the our nanocarrier. First, we synthesized the magnetic nanoparticle as a core material. Then we coated the aminocellulose layer onto the magnetic nanoparticle. And then we perform the
it's 9 o'clock.
We perform the like conjugation reaction to get the and then drawn onto the aminocellulose layer, and then finally we conjugated the PEG on the periphery of this nanocarrier, and we loaded the LCS1 drug inside this. And then we stepwise we characterize our nanocarrier, and First, we found that the size of the hydrodynamic diameter of the magnetic nanoparticle is about 11 nanometer. And after the coating of aminocellulose onto it, the particle size is increased from 11 to 24 nanometer. It is very quite obvious because there is late coating. Due to coating, the size increases. And next, again, the layer 2, again the size increased to 100 nanometer. And after the encapsulation of the LCS1 drug, the final size of the nanocarrier is 138 nanometer. And this is again the zeta potential measurement. We again it supports the stepwise conjugation. If you see this, the zeta potential of the magnetic nanoparticle is negative because of the presence of the hydroxyl ion on the surface of the magnetic nanoparticle. That is why the beta potential of magnetic nanoparticle is negative. And after the conjugation of aminocellulose onto the magnetic nanoparticle, the zeta potential becomes positive. This is because aminocellulose contains the primary amine group, and this contains the positive charge. That's why it imparts the positive charge. And again, this is due to the presence of the ester and amine. Again, the charge is changing. So it further shows that there is a stepwise synthesis of all the coating onto the nanocarrier. And then this is the thermal degradation behavior of the all the three type of nanocarrier. And we found that at about 800 degrees Celsius, there is no degradation of this MNP particle, but there is a degradation of the aminocellulose coated magnetic nanoparticle and this final coating material. That is because of the presence of the organic material onto the this magnetic nanoparticle. This further shows that there are coating present onto the magnetic nanoparticle. And then we successfully characterize our nanocarrier with the help of FTIR, and all the characteristic peaks are present in the final formulation. And we then characterize our nanocarrier with the help of transmission electron microscopy, FESM, and atomic force microscopy. And we found that the results of the size of the nanocarrier corroborate with the zeta sizer hydrodynamic diameter measured by the zeta sizer. And this is again the stepwise synthesis of the nanoparticle. And after every coating, the size increases, as shown by this AFM analysis. And after the coating, now the important point is we have coated 3 different layers of polymers onto the magnetic nanoparticle, but we have to exploit the super paramagnetic behavior of the magnetic nanoparticle so that it can be used for the magnetic hyperthermia purpose. So our question is, we really want to assess whether after 3 different coatings, still this magnetic nanoparticle retains its magnetic behavior or not. Then we have performed the VSM analysis, and we found that even after 3 coating, our magnetic our final nanocarrier contains the super paramagnetic behavior, and we can use this particular nanocarrier for loading the drug as well as for the hyperthermia purpose. This is the drug release profile, and our drug release profile shows that the controlled release at pH 7.4 and pH 6.5. And this is the uptake study, the cellular uptake study. It is very important that we have developed some nanocarrier. First, the question is whether these particular nanocarrier will be uptaken by the cells or not. So we have performed a cellular internalization study, and we have used this HCT116 colorectal cancer cells, and we treat FITC loaded nanoparticles with these cells, and we found that these discrete patches, the green color patches, because of the FITC dye, the discrete patches present inside the cancer cells and the close vicinity of the nucleus. So our nanocarrier is successfully internalized into the cancer cells. Then we performed the cytocompatibility study of the our blank nanoparticle without any drug, blank nanoparticle, to see the cytocompatibility. And we have used this HEK293 cells, which are non-cancerous cells. And we found that our up to 400 microgram per ml, our nanocarrier did not exert any kind of cytotoxic effects on the cells. And this is a therapeutic study using the MTT assay, and we found that our developed nanocarrier induced the specific killing of Bloom deficient cancer cells as compared to Bloom proficient cancer cells. After that, we perform the protein corona study, because protein corona study is very crucial, because after like injecting any kind of nanomedicine into the circulation, there's several types of various types of serum proteins present in the circulation, which adsorb onto the nanocarrier, and that ultimately decide whether the nanoparticle or nanomedicine will stay in the circulation, will reach to the tumor site, or will eliminate from the body. So this is very crucial study. That's why we performed this protein corona study. We incubated a nanoparticle in FPS media for the cell culture purpose and the human sterile serum, and we found that there's like various proteins adsorb onto the nanomedicine, our like developed nanocarrier. And we again perform that, like whether after coating this serum protein onto the nanomedicine, still our nanomedicine will be uptaken by the cells, cancer cells or not. And we found that our nanocarrier are still uptaken by the cancer cells, as demonstrated by the flow cytometry study. And then we perform the XTT study without any drug. We taken the serum protein coated nanocarrier, and we performed the XTT assay, and we found that up to the 400 microgram per ml, our corona coated nanoparticle did not exert any toxic effect. And then after that, we perform the gamma H2X study, because this is due to the inhibition of this SOD1 will lead to the increase in the ROS, and ROS will induce the DNA damage, especially DNA double strand break. So we wanted to assess the biomarker, because gamma H2X is a biomarker for the DNA double strand break, and we found that from our study that like after the treatment with the LCS1 loaded nanocarrier, there is like increased expression of the gamma H2X in Bloom deficient HTT116 as compared to the Bloom proficient HTT116. And again, we perform the we confirm our result. This is another biomarker of the DNA double strand break, that is 53BP1. And from this study also, we found that after the treatment with the LCS1 loaded nanocarrier, the increased expression of this 53BP1 increased expression of the 53BP1 in Bloom deficient HTT116 cells as compared to the Bloom proficient one. And finally, we performed the cleave caspase 3 as a marker of the cell death, and we found that after the treatment with the LCS1 loaded nanocarrier, the particular cells, Bloom deficient HTT116 cells, exhibited the enhanced expression of the cleave caspase 3 as compared to the Bloom proficient HTT116. So the conclusion of my study was customized triple polymeric layer coated magnetic nanoparticles was synthesized and characterized for the encapsulation of LCS1 drug, which is a specific inhibitor of the SOD1 enzyme. The polymeric coatings onto magnetic nanoparticles reduce the cytotoxicity of bare magnetic nanoparticle, because bare magnetic nanoparticle is comparatively toxic as compared to the polymer coated nanoparticle. And the final LCS1 loaded nanocarrier showed enhanced therapeutic efficacy from 61 efficacy enhanced from 61 to 104 times as compared to free LCS1, which selectively targeting cancer cells having Bloom defects. So I would like to acknowledge my collaborators, my lab members, and my funding agency, and obviously I would like to thank to the ICBH for inviting me for the oral presentation.
Thank you very much for your valuable presentation, Dr. Khan. Thank you. Is there anybody from the audience who wants to ask a question or who wants to contribute? Yes, I think it's a bit late, and yes. Okay, it was a very nice presentation. Thank you very much, Dr. Khan. So if there isn't any question, yes, Dr. Ajaros.
Dear Professor Dr. Şebnem Alanyatosun, thank you very much for your valuable contribution. It was very nice moderation of you. And
I appreciate it. It was my pleasure.
thank you very much again for your contributions. And I think we can close the session today. And dear respected participants, today we are closing the session. Tomorrow is our last day, and we will be happy to meet you tomorrow again, and see you tomorrow.
Okay, see you tomorrow. Thank you very much.
Bye-bye.
Bye-bye.